This part is going to be about early clinical development. Determining the structure of a protease was crucial to the development of inhibitors that might render the enzyme inactive and thus prevent the virus from maturing. In 1992, because Crixivan had an acceptable toxicity profile in animals, Merck decided to proceed to safety trials in healthy volunteers not infected with HIV. In February 1993, the 12-day 12-patient safety study with Crixivan in healthy volunteers not infected with HIV at multiple doses began at Thomas Jefferson University Hospital in Philadelphia. In April 1993, Merck and 14 other pharmaceutical companies announced the formation of the Intercompany Collaboration for AIDS Drug Development. The companies agreed to work together to facilitate, through the sharing of information and drug supplies, early human trials of combination drug therapies for the treatment of HIV and AIDS. In June 1993, Thomas Jefferson University Hospital and the University of Alabama, Birmingham, started a 12-day proof-of-concept study of Crixivan in HIV-infected patients, which would assess safety and give an indication of antiviral activity. In October 1993, there was a 6-week with 60-patient safety and efficacy trial of Crixivan using doses of 200 milligrams and 400 milligrams every 6 hours. 600 milligram dose every 4 hours was added later during the protocol. Information on the pharmacokinetics of Indinavir in humans has been obtained from a total of 25 studies. Data from 364 subjects was presented, of whom 179 were HIV patients. The vast majority of the subjects included were male, 84%. One study was actually performed in subjects with mild to moderate hepatic impairment. Indinavir is rapidly absorbed in the fasted state with an absolute bioavailability of approximately 60% within the dose range of 400 to 800 milligrams. The bioavailability of a single 800 milligram dose was approximately 65%. Administration of a 400 milligram dose with a high-fat breakfast reduced the relative bioavailability to approximately 20% compared to fasting conditions, but bioavailability was unaffected when an 800 milligram dose was administered with a light meal. We must also mention that no major pharmacokinetic differences were identified in Caucasians versus Black or in HIV patients versus healthy volunteers. With the development of the low capsule strength, the pharmacokinetics of Indinavir have been studied in a total of 95 children and adolescents aged 3 to 18 years old, of which 70 received the recommended dose, which was adjusted from calculated body surface area. No study has been performed in patients with renal insufficiency, and there are no pharmacokinetic data either for subjects older than 60 years. Combined results of these studies showed that the chosen dose of 800 milligrams every 8 hours exerted an optimal antiviral activity. Moreover, higher doses increase the risk of adverse events such as nephrolithiasis and hyperbilirubinemia.